Today, it's great to have Derek Reiman giving a research at TTIC talk. Derek is a research assistant professor here, and so every Friday we have a talk by one of our faculty, kind of letting everyone know about the different kinds of research going on here. And so Derek works in computational biology, and in particular, a lot of things have to do with the microbiome, the gut, all the interesting things going on. In your gut. And today, talking about modeling host microbiome interactions at the metabolic interface. Let's take it away.
Thank you, Avram. So like Avram said, I work a lot with microbiome and immunology, so maybe think of it like it's a computational pathologist, even though that's not really a term yet. But today I want to talk about kind of microbiome studies, and in particular, kind of focusing in on a topic that's really interesting to me, and that's looking at kind of this metabolic interface between host and microbiome. So for those who have seen my talks, you've probably seen the next few slides about the microbiome, but for those who have not, this might be something new. So the microbiome, when we talk about it, that kind of it's a collection of microscopic organisms. And generally we talk about this in terms of bacteria, but we can also think of fungi, protozoa, and viruses to be part of this community as well. And it's believed that this environment functionally interacts with its host at the environment. So in the case of kind of clinical microbiome, we talk about the human, or maybe mouse models, but you can also think of marine biology, so the microbes in the ocean, agriculture, the microbes in the soil. And in the case of clinical microbiology, different things can impact this community, such as genetics, dietary intake, the environment you live in, your lifestyle, and hormones. And there's also different communities that we can study. For example, there's the nasal microbiome, the oral microbiome of the mouth, lungs, stomach, the intestines, sex organs, and skin. In particular, I focus on what's called the gut microbiome. So that will be the combination of stomach and intestines. So the reason why I'm really interested in the gut microbiome, and it's become such a hot topic in the last few years, is our kind of growth in its understanding of what it's doing for us in a health. So some of the earliest studies really looked at the role of the gut microbiome in metabolic diseases. So we know a lot of metabolic processes go on in the intestines, and so the microbiome is a heavy component of that. And under healthy circumstances, you generally have healthy phenotype or some sort of health status, so you're healthy. But if you get some sort of pathogens or some foreign particles that your body doesn't like, you can start to signal inflammation, and that opens up these tight junctions between cells to create leaky gut. Microbes can go out, these molecules can go out, and you can start to see things like IBD or inflammatory bowel disease, obesity, insulin resistance, and all sorts of nasty things. We also know that the gut microbiome is incredibly important in the development and modulation of the immune system. So from infancy or even before infancy, the composition of this microbiome is responsible for kind of the training of this naive immune system in infants. And in turn, as that immune system kind of learns and develops, it's responsible for kind of protecting the gut microbiome. And when we can see dysbiosis in this kind of stage of life, we start to see things like early autoimmune disease as well as food allergies. An interesting topic that kind of came around in 2020 was the understanding that the gut microbiome is also connected to your brain. This is called the microbiome gut-brain axis. And so if you ever kind of been stressed, you might remember that sometimes when you're stressed you get a stomach ache. But it also works in the other direction, where microbes in your intestines can actually be used to help reduce stress. And so that's a whole kind of therapeutic route people are exploring. And then the most interesting and most recent is its role in cancer immunotherapy efficiency. So we've kind of discovered around 2019 that the gut microbiome plays a significant impact in cancer outcome or response to treatment. And so in a clinical trial in 2022, one of the first that actually introduced kind of these live biotics into cancer treatment, we've got two arms of treatment that receive this frontline immunotherapy treatment, nivolumab and ipilimumab, which are both checkpoint inhibitors. With the difference being that one group, shown in gray, received a live biotic which had Clostridium butyricum. So this produces butyrate, a short-chain fatty acid. And what happened was that without the presence of this live biotic, after about 7 months, all these patients had progression in their cancer, meaning the treatment did not work. We had to switch to a new line of therapy because the cancer was growing. However, with the introduction of just this microbe, even after 24 months, so about 2 years, about half the patients were still progression-free. So we were seeing either reduction in cancer or it wasn't growing.
Yeah, so this is checkpoint inhibitors being done in both cases.
Yes, so the actual checkpoint inhibitor, the drug therapy is the same. The only difference is this microbe being introduced. So hopefully with that, I've kind of convinced you that of how important the role of the gut microbiome is in human health. A couple other things that we don't talk about is its role in cardiovascular disease as well as inflammation. But some of the challenges in the clinical setting, especially with human microbiome, is the inter-individual variability. So my gut microbiome is very different than yours, which is very different than yours. And so kind of identifying what part of the microbiome leads to kind of this dysbiosis, development of disease, it can be very challenging. But because it is, there's large plasticity in it, meaning we can change it readily and easily, it is becoming a kind of a common targetable factor for therapies. So how does the microbiome function? And so kind of the center of this talk is this interface to where we know the microbiome of the gut interacts with you at a metabolic level. And so, for example, you can, depending on what you eat, what you intake, what you're exposed to, your gut microbiome will interact with you through metabolites. And we'll talk about metabolites here in a few slides, but you can think of them as just small molecules floating around, either in the cell, the bloodstream. And the way that it can interact is either through producing its own microbial metabolites and introducing those into the environment, altering metabolites from the host, or through co-metabolism, requiring a metabolite from the host and metabolite from the microbiome, and creating some new interaction. Okay, my clock is right. Okay. So to kind of hone in on just how impactful this interface can be, I want to talk about some work I was lucky enough to collaborate on with Alexander Chervonsky's lab at U Chicago. And so this was a very interesting study to where we looked at, they took what are called NOD mice. So these mice are genetically disposed to develop type 1 diabetes. So we'll talk about two cases, one germ-free. So these mice have no microbiome, and these mice have some conventional microbiome with no pathogens. And in both cases of the germ-free and the microbiome ones, when you feed them a normal diet, shown in the blue, over about 30 weeks, they will develop type 1 diabetes at about 75 to 80% rate. So most of them will develop type 1 diabetes. They're just genetically predisposed to it. However, if we change their diet from a standard chow mouse diet and we feed them either hydrolyzed casein or intact casein, they do not develop diabetes. So something interesting happened. So we've removed diabetes from this equation for now, and now we're going to introduce a new component, gluten. So when we add gluten to this equation, for the mice that don't have a microbiome, we don't really see a change. They are still have no diabetes, it's virtually gone. But when we look at the mice with the microbiome, compared to kind of our baseline chow blue, our normal casein where we remove type 1 diabetes in the red, when we look at the black and white circles, which are two different gluten sources, we now have diabetes again. And so something happens between the microbiome and gluten to bring type 1 diabetes back. So through some biological experiments that I won't go into, they identified E. faecalis as this microbe that can digest gluten. It has a protease which will cut the gluten, and through cutting the gluten, it creates a new metabolite, which when introduced into the system, the host system, elicits an immune response, an autoimmune response leading to type 1 diabetes. And what we can show is if now we take these gluten or these germ-free mice, so there's no microbiome, and we give them casein and gluten, and this microbe, this one single microbe that we suspect causes diabetes, yeah, we get diabetes. But if instead we either take a knockout mutant of this mouse, so we eliminate the mouse's ability to respond to this molecule, or if we mutate the microbe and remove the ability to cut the gluten, no. And so what's the moral of the story here? We don't eat gluten. No. The moral is that even on kind of these simple scales, host microbiome interactions can have a huge profound on health. So hopefully with that, I've convinced you of kind of the need to really study this in clinical settings. And so we're going to be talking about how to model kind of gut microbiome and metabolomic data. Really kind of considering 3 questions that are of interest, which when we look at kind of disease states in clinical settings, which metabolic pathways are being altered by the microbiome, which microbes are responsible for these alterations, and then how do these alterations lead to differences in health. So we have 2 different data types that we need to talk about. So how do we collect and present gut microbiome data, and then how do we present metabolomic data? So to quickly kind of go through this, when we want to study the gut microbiome, we generally need to take a microbiome sample. So for gut microbiome, this will be either fecal, or you can, if you have a biopsy, you can take cecum. And we take microbial DNA. So we weed out the human DNA, we keep the microbial DNA, and in one route, which is called amplicon sequence variant, or amplicon sequence variant sequencing, we isolate a specific gene of the microbiome genome, which is going to be the 16S rRNA gene sequence. Every microbe has it, so we can confidently grab them. And then we can cluster these sequences into operational taxonomic units, map these OTUs into kind of this taxonomic database to where we can now identify within each gut microbiome sample which microbes are present, how abundant each microbe is present, as well as some sort of phylogenetic information regarding these sequences so we know who's present and how similar they are. The other route, which is a little bit more expensive, is rather than taking just the 16S data, we're going to take all the microbial DNA. And again, we can map these genes to reference genomes and get the same information. But we can also look at these genes, since they aren't just the 16S, but all functional genes, to some sort of pathways and get a new layer of information for functional enrichment. For the topic of this talk, we're only really going to be considering the top one, the microbial abundance. So who's present and how much of each microbe is present in that sample.
How many of these microbes are any of these microbes human specific?
I would imagine there are. I know there are like mice specific. I would imagine there's also human specific, but generally mice specific ones are labeled specifically like with mouse in the name. I've never seen one with just human, but I would imagine since it's kind of like the standard that they would be. Now it doesn't mean you can't like take a human specific one and put it in a mouse, but you might generally not see it grow naturally. So the other component of modeling these interactions is understanding of the metabolome. So metabolomics is the identification of small molecules involved in metabolism, which are life-sustaining chemical reactions. And these can be all sorts of different types of compounds. Some of the more common ones that you might hear of are vitamins, hormones, fatty acids, sugars. This actually provides, if you're familiar with biology, in the central dogma, you know kind of genome or DNA, RNA, protein, but this kind of adds a layer just after that between what we observe in a phenotype and what we can observe in the proteome. So in order to measure the metabolomic data, we generally need a sample once again. So this can either be blood, serum, or actually I don't think you would use blood, but it could also be fecal matter in the case of gut microbiome. You perform some sort of chromatography and mass spectrometry. So for example, you can do liquid chromatography and mass spec, and you get data that kind of looks like this 3-dimensional graph here, where we can stratify compounds based on mass over charge, so M over Z based on mass spec, and retention time based on chromatography. So you have this XY coordinate plane of mass charge and retention time. And then we have these intensities or these peaks. And so the height of this peak can be some sort of ion concentration or signal for that feature. Now, because we are just looking at kind of M over Z numbers and retention time numbers, you don't really know what you're looking at a lot of times in untargeted. So basically we're casting this very wide net, not worrying about what we're capturing, and we're hoping to maybe identify it later. So some of the history behind the modeling of microbiome metabolome interactions kind of started early in the 2010 with a colleague of mine, actually Peter Larson, who kind of put forth this model called predictive relative metabolic turnover. And then additionally, another commonly used method is constraint-based flux balance analysis. And in both of these, the main drawback is we have to completely rely on a priori knowledge, meaning if we've got some gut microbiome sample, we need to know two things. First is we need to know for each gut or each microbe that we observe, what sort of enzymes can they give us based on some sort of annotated reference that we can pull from. So not directly looking at what we're observing in the data, but based on some prior annotated data. And then two, we need to be able to construct our reactions. So if we have some sample and we can put together some pathway, we know this is generally the structure of the pathway that we want to model. We can perform flux-based or flux balance analysis to kind of learn the fluxes of the reactions to achieve the steady state. However, the drawback here is since you're relying on everything that is already previously known, it's really not possible to learn anything new about host microbiome interactions. But within the last few years, we've seen the emergence of paired metabolomic microbiome data. So now people are not only sequencing the microbiome, but they're also sequencing the metabolome from the same sample. And that actually gives us an ability to start looking at data-driven approaches. And so, for example, if we have some gut microbiome, we can do some machine learning, for example, some neural network, and we can predict the metabolome. We can do this in a multivariate learning, and then try to do some interpretation to figure out how it's making its prediction and what's going on, and it will look at functional clustering. So this is kind of a general sketch of a model that we developed back in 2020 called MIMEnet. So MIMEnet was one of the first data-driven approaches for kind of exploring these host microbiome interactions at the metabolic level. And so it requires paired data, which was kind of being generated at the time and continue to being generated. And you do some standard pre-processing, filtering, and compositional transformation. And it's a pretty straightforward model to where we're going to predict our metabolomic composition from our microbiome composition. And from that, we can predict different metabolites. We can use this model to infer interaction matrices. And then based on behaviors of metabolites and microbes, we can do some clustering and network construction. And we'll go through all this step by step. So in the first step, we have kind of it's a simple model. Neural network, it's basically translating your microbiome composition to your metabolomic composition. And we're going to do this 10 iterations with 10-fold cross-validation. And we need to do that because we need to be able to evaluate different metabolites. So for example, if we train our model and we have metabolite A and B, and metabolite A has some Spearman correlation of a decent number and B is a little bit less, we kind of have a problem now is how do we determine which metabolite predictions are significant? Because the key here is that because metabolomic data is such a wide net, everything we've captured is not guaranteed to be related to the microbiome. Microbiome is probably only associated with very few metabolites and metabolic functions going on. And so how do we determine which ones are actually able to be predicted well enough by the microbiome to be associated with it? And we do that through kind of a model bootstrapping approach. So we're going to shuffle our metabolomic data, and we're going to do 10-fold cross-validation. We're going to do it 100 times. And luckily, microbiome data is small enough that this actually doesn't take very long. And that gives us an ability to create some null distribution, assuming that there are no microbiome metabolomic interactions shown in blue, and we can set some threshold. And based on what we observed, such as shown in orange, we can start to identify which metabolites are significantly correlated with our data, our microbiome data. So now we've kind of done the first step of identifying which metabolites are associated with the microbiome composition. So next we need to identify how these microbes are being related to the metabolites that we've pulled out. And so we actually use a kind of an older method that which is quite quick, since we have to do it many times, and that's just Olden's method of feature attribution. And so if you have a neural network model, you represent each layer as some sort of matrix. If you perform matrix multiplication across the layers, you get this final matrix where each row represents an input feature and each column represents an output feature. And so for example, if we look at I and M, this represents some sort of interaction between microbe N and metabolite M. And so now we've got a way of kind of connecting our input to our output in a meaningful biological way. And again, we've done this 100 different times. We have 100 different models. We have 100 different matrices. And this is important for the next step because we have the assumption in microbiome data that groups of microbes affect groups of metabolites in similar ways. And so kind of this one-to-one identification doesn't really make sense. And so we're going to want to cluster these. And so we take our interaction matrices that we have 100 of, and we perform consensus clustering for the microbes and metabolites independently. And so that means is for each interaction matrix, we determine a number of clusters K, we determine a connectivity matrix, and we can do that for all 100 of our matrices and get the mean connectivity matrix. We can then plot the cumulative distribution function of our connectivity, our consensus index. And so ideally a good consensus would be a lot of zeros and a lot of ones, so you want to see a sharp rise. And so that's what we see as we increase the number of clusters. We see our clustering get better. And so in order to evaluate kind of the improvement we observe, we're going to approximate the area under the cumulative distribution function using rectangular approximation, and then calculate some sort of proportional area change between k-1 and k. Now the issue, since we're using hierarchical clustering, this will always increase. And so we do set some threshold where if it doesn't improve by a certain amount, we actually do not increase the number of clusters. And so with this, we actually have everything we need to build our final model. We have the metabolites that are associated with the microbiome, and we have a good estimation as to the number of microbial modules and metabolic modules. And so we can train our final model, which can then be used for some external validation. We can perform bi-clustering using our predetermined cluster numbers. We can create some sort of modules based on these clusters, as well as module-based features, looking at the kind of average abundance of the members of whatever that module is. Try to speed up a bit. So we'll do a case study. We look at IBD. So this is a nice study because it has two different data sets. It's got PRISM, which is from the Massachusetts General Health, as well as a validation set, which comes from the Netherlands. But the nice thing is that both these data, even though they're demographically different, were sequenced by the Broad Institute, so there is no technological variation. All the variation is biological. And we compare our model to the previous the first model that kind of came out for data-driven approaches, which was MelonPan, which was an elastic net model, looking at just of the 8,000 metabolites in this data set, only about 440 were annotated that we knew what were. So we looked at the performance on those, primarily because MelonPan models one metabolite at a time, and so this took about, I think 2 days for MelonPan just to do 400. And we noticed that something strange happened, is that there was about 200 of these metabolites that MelonPan just could not model at all. And so our hypothesis based on this observation was that by learning all the metabolites at the same time, we know metabolites work together in pathways. They have some sort of correlation between their behavior. So is there some sort of transfer learning going on between our outputs? And so we evaluated that kind of in a simple study to where we kind of run MIMENet in two different fashions, one using just the identified metabolites and one using the full set. And we only evaluate the identified. So we want to know how are these identified purple ones changing? And we do that under increasing levels of noise, added to the identified training metabolites. And so what we observed at baseline is that with the additional 8,400 metabolites, there is actually a significant increase in performance from an average correlation of 0.26 to 0.31. And that under increasing levels of noise, we can see that if you're just using the annotated metabolites, which are undergoing the corruption, it quickly deteriorates and the model collapses and breaks. However, even under extremely high levels of noise, to where essentially all signal is gone in these annotated metabolites, using the rest of the set is actually able to maintain some sort of decent level of correlation of using transferred knowledge. So back to IBD, we kind of did a case study where we clustered. We also observed that using MIMEnet modules, so the features that we can construct using our clustering, had the same level of prediction performance for the disease state compared to just using the original models or the original features. And this was interesting because MIMEnet actually never is given information about disease state. We can further, upon post-analysis, identify specific microbial and metabolic modules related to kind of healthy patients and diseased patients in this IBD study, as well as identify things that are commonly known in literature with IBD, such as short-chain fatty acids being associated with health, generally in all diseases and related to gut microbiome, as well as things like secondary bile acids being associated with healthy patients, which is really informative because we know in IBD, an IBD patient loses the ability to deconjugate conjugated bile acids. So bile acids generally go from primary bile acids, go through some sort of conjugation, and then have to be deconjugated into secondary. So that process is actually halted, and it actually captures it. And so now we have a model that can start to connect the dots between microbiome community compositions and metabolomic compositions. So one, and this has kind of led to a lot of collaborations and use. And one thing that kind of rose in some of these collaborations was that can we apply MIMEnet to other interfaces other than metabolomic? And the answer is yes. And this comes from a work by Catherine Amato at Northwestern, where they were interested in kind of the interaction between the gut microbiome and gene expression. And so what they're studying is kind of a difference in brain prioritization. So in primates, there's kind of two different ways of brain growth, one being high prioritization, such as humans and squirrel monkeys, to where, especially in the early stages of life, the body is providing a lot of energy and metabolism to sustain rapid brain growth. And the other, such as a macaque shown in the bottom, is non-prioritized. So they have a much lower energy requirement, but their brain kind of grows at a much slower but more steady rate across their life. And their kind of hypothesis was that the gut microbiome was actually being selected for in order to produce these short-chain fatty acids, which in turn promote gluconeogenesis and kind of production of glucose for the brain. And so generally a standard approach would be something like gene expression analysis, and you do some sort of differential expression analysis. And we did that, and we found that compared to the squirrel monkey and human versus the macaque, very similar behavior in the gene expression. But we can also take the gut microbiome and look at the gene expression. And we notice very similar patterns, especially with cluster 0 and 3, focusing on this metabolism of lipids. And this is important because we know that these lipids have to be metabolized to help produce the energy requirement for this brain growth. And that kind of gives this new perspective of the model to where we know that there's this interaction between the gut microbiome being selected for the expression of genes in the liver and the production of energy for the brain that creates the cycle. Yes?
Sorry, you might have mentioned this, but gene expression in which cells?
This is just RNA-seq. So this, liver.
What tissue?
Liver. Yeah, sorry. Okay, so back to IBD. That was just kind of a detour. So one of the things we observed when we did MIMEnet was we observed that the prediction performance of the metabolome was quite a bit better than that of the microbiome. And this is interesting based on kind of our understanding of the kind of relationship between the metabolome and the microbiome, each being kind of influenced by separate factors, a lot of overlapping, some kind of unique to each modality. And so we had the question of, is this always the case? Is it always that the metabolome is more informative? Are there some situations where the microbiome is more informative? Or kind of more importantly, can we actually like combine these two into some sort of meaningful functional representation to even further produce or further improve our prediction performance? And so this led to some work that was done by Tina Khage, who was my intern over the summer, and kind of continued working throughout the year, and still ongoing. But we kind of have two aims for this project. And the first was we want to learn how to integrate microbiome and metabolomic data into a single representation to be more predictive than each modality independently. And this is kind of an extension from some earlier work by Tina, 2021. And so what we did was we took our microbiome data, and so we kind of did an exploration analysis just to kind of look at what it looks like. This is our IBD data set. I kind of stick with this one because it's clean and nice. And we look at things like alpha diversity, beta diversity, and everything kind of makes sense health-wise from what we understand as the microbiome. We know that diversity, healthy patients generally have a higher diversity, so we see in the control patients alpha diversity is increased, and we see a nice clustering in the beta diversity, which is our way of data visualization in the microbiome field, where the healthy patients shown in red are kind of more pulled towards the left. And so based on some our observations and some previous work by myself where I've kind of modeled just microbiome data independently, showing that the use of latent representations of autoencoders can help with things like forecasting and phenotype prediction. We want to see if we can take kind of the same approach here. And so we want to kind of compress our metabolome and microbiome into a single representation that kind of captures the essence of both. And we need to do that in a way that we have kind of two models in one. And so we have two sets of inputs, metabolome in blue, microbiome in green, and we also have two ways of treating our outputs. So we've got these metabolomic reconstructions, which need a specific activation function. We're going to be using linear. And we've got microbiome, since we're using compositional, these have to be softmax. That means we also need to kind of decompose our cost function to where we can have something like the mean squared error for our metabolome and some sort of KLD or Kullback-Leibler divergence for our microbiome. We can balance those with some alpha coefficient, even though I believe in our results that I'm about to show, we just use 0.5, it's equally balanced, and some sort of regularization. And what we see, and then what we can do is using our latent representation and our original features, we can compare some sort of prediction. For example, in this case, we'll be using a neural network with standard cross entropy loss, what we see is actually in both, when we're looking at a binary disease stratification, so healthy versus IBD, as well as multi-class, so this has IBD is split into both ulcerative colitis and Crohn's disease. Two things that occur. One is that we observe in each case that the combination of the microbiome and the metabolomic features, whether they're concatenated or compressed into the representation, have the greatest predictive power. And two is that in each case from data modality, using the raw feature compared to a latent representation, using the latent representation improves performance, whether it's just microbiome, just metabolome, or the combined profile. And that kind of correlates with a lot of previous studies that I've done. So now in the last, I guess we're good on maybe 10 minutes, I want to talk about kind of the next direction of this project. So this is purely conceptual stage, so I might get a little overly excited and talk fast, but it's addressing a big problem in microbiome studies. So say we've looked at kind of how to predict some sort of disease from microbiome data, but you want to build some sort of generalized model. I'm going to bring in all these data sets because the growth of microbiome data has become exponential. We've got tens of thousands of studies and data sets available, and you want to study colorectal cancer. So you grab these 7 data sets and you want to learn some generalized pattern in disease development to understand colorectal cancer better. So you take your model, and we're going to ignore the blue for a second. So you just take microbiome data, you're compressing it, and then you make a prediction and it sucks. It's terrible. And then you look at your latent representation or your data and you realize it's because your data has a massive amount of study bias. And this is a big challenge in microbiome data, because if you look at these 7 colorectal data sets and you independently do an analysis on each one, looking at which microbes are related to colorectal cancer, when you look at the overlap of these informative features that are in at least 4 of these studies, there's only 5. In at least 3, there's 8, and any more than 4, any feature shared by 5, there's none. And so when we look at these data sets, a lot of this challenge comes from the fact that the features don't overlap. And so when you look at these taxonomical annotations, there might only be a 20 to 30% overlap. And so your options are either to include the union, which you'll get a data that looks like this, where it's very biased based on the study, because microbes are only present in one data set, or you can take the intersect, in which case you'll be left with almost nothing, and you leave all your signal. And the reason that you leave all your signal is because we know, based on previous studies, that different microbes can lead to the same function. What does that mean? So say we have 3 different data sets, and we have data set 1, 2, and 3, and there's some sort of disease data sets, and we know that basically on truth, these are all related to this pathway, this metabolic toy pathway that I've constructed. In data set 1, these microbes are responsible for some interaction to where they cut this reaction from A to E. And so you don't generate E or F, but what that means is that there's more A going to B and then C and D. So you see elevated amounts of B, C, and D. You could also see data set 3, which has maybe a different interaction with this pathway, but maybe it elevates the metabolite of B, maybe it produces B on its own. And so since you have more B, you'll see more C and D. And maybe data set 2 is actually a different disease or not related to this pathway. And the disease is actually caused by an elevation of C. So in this case, with just microbiome data, there's really no way of comparing the function of the microbes from data set 1, these 3 green microbes, and these 4 microbes of data set 3 in any functional way. However, now we have metabolomic data. So let's bring that into the picture. And we've captured bits of our pathway in our metabolomic profiling. So say in data set 1, we observe A, C, and D. Data set 2, we observe A, B, and E. And in data set 3, we observe A, B, C, and F. Well, now we do have a way, because we can associate the microbes from data set 1 with these metabolites in A, C, and D. We've seen kind of how to do that looking at MIMEnet, and there's a couple other tools that facilitate that. We can associate the microbes in data set 3 with the A, B, C, and F. And since we can associate those metabolites to this overall pathway, now we know the function of this pathway. We can now connect the microbes from data set 1, the microbes from data set 3, and have a functional way of doing that. And so now we can go back to this model to where you've trained it, and say you look at this, your latent space, and it's incredibly data set biased, and this first node just represents the first data set. So our hypothesis is that if we use something along the lines of some sort of discriminator network and train it adversarially, for example, to predict the data set, we can pull the latent space to actually be, or to share the same distribution across data sets. So kind of have this adversarial training, so we remove this bias. And then we can inject our data sets back there, so we know how to go from our latent space, which now shares a distribution, back to our data set specific distributions, with the hopes of, instead of capturing a data set, now we can capture similarities or functional similarities across these pathways. Because now it can use this node to decode data set 1 using this green microbes, A, C, and D, data set 2. To actually get the metabolites, you don't get the microbe because the microbe is not associated, and then data set 3 to reconstruct that. So in principle, we kind of have So this actually allows us to answer 2 very important questions. One is we can now look at does this generalized latent representation, which hopefully captures some sort of functional structure, produce or improve some sort of prediction performance in a meta-analysis study. And then two, which is a very interesting question and would be very cool if we could answer, is what are the different microbes across different data sets which are doing the same thing related to disease? And so this is kind of leads into our aim two. So we can have this kind of adversarial structure where we have an adversarial discriminator which can predict the data set. We have a decoder which just focuses on reconstruction, and then an encoder which actually does the reconstruction as well as is trained adversarially to our discriminator. And we've pulled in a couple new data sets to kind of train this. We have 5 data sets of paired metabolomic microbiome data with different diseases, so colorectal cancer, IBD, and end-stage renal disease. And we're currently in the process of developing this out. So the next steps for this project are, well, we've already done the exploratory analysis on these new data sets. I'm not going to show that today. We're currently actually going back and looking at aim 1 at these 4 new data sets and seeing, do we see the same behavior? So in each of these other data sets, do we see the behavior that the metabolomic profile is more predictive than the microbiome, and the combined profile is more predictive than both, and the use of an autoencoder improves even more? So that's kind of in progress still. And then hopefully this spring we can start developing the adversarial model. But I thought I'd talk about it just to kind of maybe garner feedback or interest, because I'm really excited about it. And so I'll actually end there, and I'd like to kind of acknowledge my collaborators at UChicago, so Ali Khan, who I collaborate on a lot of work with, and then Alexander Chabansky and his student Matthew, who has defended successfully and moved on, who were responsible for the mice getting diabetes, as well as collaborators at UIC, Dr. Brian Layden, who works a lot in endocrinology and diabetes, and then my former advisor, Yang Dai, and then her student, Tina, who was my intern and instrumental in a lot of this work, as well as Catherine Amato from NYU, who did the brain study. So I will end there and take questions, and thank you all.
Yes. So I guess maybe one question would be, excuse me. So my understanding is you had that neural network that was going from what? It was going from the microbiome to predicting the metabolome, basically. So and then from that you were predicting whether or not people would have IBD. But then did that prediction incorporate?
So which neural are you talking about? This one or the?
No, the earlier one, the first neural network, I think. I guess the question was whether or not it incorporated the fluxes at any point, or if it only cared about the microbiome abundances. No, this one. Yeah.
This one. Okay. So this model actually never you're talking about so there's no fluxes in here. We're flux balance analysis is kind of the a priori method. So this is data driven, and we also don't show this model disease state ever.
Right.
So you never predict disease state, right?
So what we do at the end is as a kind of post analysis experiment. If we So for example, if this was our clustering in our network, we designed features for each of these based on our clustering, and then we use those features to predict disease state to see, does it share the same level of information as the raw features? Is it organized in a meaningful way?
Maybe a related quick question about that is could you construct the matrix you need to do flux balance analysis from this interaction matrix, or are they fundamentally different things?
I would say they're fundamentally different, but it might be interesting to see if there's some sort of, not like a full mapping, but if you can leverage the information from the interactions to kind of help maybe learn from in a flux balance analysis something new. Because a lot of times with flux balance analysis, your reactions are kind of hard coded in, but maybe you can start to like learn new reactions or.
I guess for the new work that you're doing, where you have multiple different data sets, but you kind of want to get to this question of, do they encode the same information? Have you considered any contrastive approaches? Because that seems to be like a very popular direction for paired data sets, right? Getting at the same function across different samples.
We haven't, but it's actually a good idea. Probably have to go back to the drawing board, or we'll see how this one goes. Maybe compare, like, could be another model, and we'll see which is a better approach. Yes.
So one criticism people often have about stool metabolomics is that you're only getting like the final product, and a lot of metabolites are actually taken up by the epithelial cells in the gut, so they never make it into the stool. And I wonder if you've built this matrix of interactions, but have you tried building something similar for like a cultured metabolomics, where you just have to cut the microbiome, feed it some diet, and you get the metabolomics out? And I wonder how different versus similar that would be from this, right? So you could try
Like in like talking about like a culture dish.
So, I mean, people have like bioreactors where you can construct a microbiome that's based on some human or mouse or whatever you want, co-culture them, give them some standard American diet, basically just the microbes, and then measure the metabolomics, where it's thought that you can kind of remove that factor of the epithelial cells taking up the short chain fatty acids or whatever. I think that could be really interesting to kind of see if you can detect what's the difference between that matrix versus what you would have seen in the culture, because I know those data sets exist.
Yeah, I think that'd be really cool. Like that would kind of give a perspective of, what are you losing along the way, I guess. And
Exactly.
but I guess the challenge might be you also lose the perspective of how for example, like host cells and proteins from the host cells, which might be also modifying what you see. You're gonna be losing that as well. But it would be a very like microbiome specific function.
Well, or you might be able to see what you would expect, like what you can see where does the host come in, what does it change, I guess. And also, you mentioned in the beginning something about blood metabolomics. So there is blood metabolomics. I was wondering if I think you should look at it at some point, because it's actually
everything I've
really
used is
interesting.
serum or fecal.
Yeah. Okay. Yeah. Sorry.
What's the benefit of blood over serum?
So blood is actually, so in some IBD colorectal cancer, you can see some of the blood metabolomics, like with the leaky gut, making it the gut metabolites making it into the bloodstream faster than if the gut was like the epithelial integrity was like there. So you can see some changes there, but I don't know if this I assume this method would detect that very well.
I'll have to check out the blood metabolomics. I haven't seen that yet.
I have a question. So actually, earlier in your talk, you had this example where there was a cancer treatment that was really helped a lot by the presence of microbes. What was going on there?
So in that particular case, I got to go back because it's written in my notes because I don't have it memorized. So this CBM 588 has Clostridium butyricum, and so what they did is this microbe will produce short chain fatty acids, in particular butyrate, which is like the talk of the town. And so when they introduced it into the microbiome, their goal was to help promote the growth of bifidobacterium species. So this microbe is actually introduced not to interact with the drug, but to grow microbes that will help with the drug. And so they see the growth of this bifidobacterium species, and we don't really know why. There's a lot of research going into why. And there's a couple different hypotheses as to how these microbes, whether they are interacting with the drug itself to sometimes suppress it, or stimulating the immune system to kind of promote the use of this drug, whether they're actually translocating through leaky gut or if it's just the metabolites. So there's still a lot of research going into this as to why. It's just really cool.
The checkpoint inhibitor is unleashing the immune system in some sense.
So yes, the checkpoint inhibitor. nivolumab, I believe, is PD1 checkpoint inhibitor, and ipilimumab is, I believe, CTLA4. And so what happens is the cancer is really good at kind of using these pathways, which essentially, if you have an immune system that's activated, it's pushing the brakes. So if you're driving a car, you're pushing the brakes, you're slowing down. And so these drugs actually take your foot off the brakes and allow the immune system to keep going. So they prevent it from being stopped. So in this case, it's facilitating the efficiency of this drug.
So the immune system is presumably more active in the presence of the microbe.
So that would be one hypothesis. A lot of people think that the presence of the microbes are facilitating immune response. But again, we still don't fully know.